Welcome to another session. In this session, I have a talk about different aspects of linkage, the meaning of linkage, the importance of linkage. What is the meaning of linkage? Linkage means the association. Linkage means the attachment. That's why they move togetherly. In case of genetics, in case of gene, if the some of the genes move togetherly from one generation to another generation, which is known as the linkage. So what is the exact meaning of linkage? Let's start. Suppose that this is the one pair of homologous chromosome, and here one chromosome which contains capital A and small b, two alleles, and another chromosome which contains capital A and capital B alleles. At the time of gamete formation, this chromosome enter into one gamete along with capital A, small b, and another chromosome which enter into another gamete along with capital A and capital B. It means that the capital A and small b, they are associated to each other. That's why at the time of inheritance, they inherit togetherly, which is known as the linkage. And here also capital A and capital B, both the genes, they are present on the same chromosome. That's why at the time of movement, along with the movement of the chromosome, these two genes or these two alleles, capital A and capital B, move togetherly or enter into the same gamete. That's why A and B are linked to each other. This is known as the linkage. Suppose that the same chromosome which contains capital A and the small b, and they are present far distance from each other. And in another case, suppose that here the two genes present very close to each other. So in this case, the linkage, the strength of linkage, which is weak, but in this case, the strength of linkage, which is more or which is very strong. Why? If the distance between two genes is more, then there is the chance to break in any place in between A and B at the time of crossing over in the meiosis. So suppose that here the gene present far nearer to than this chromosome, and here another chromosome which has the two genes nearer to each other, and here two genes which are close to each other. The strength of linkage is more here. The strength of linkage here, it is more here because they are very close to each other, and the strength of linkage is very much strong than this one. And here also the strength of linkage is more than this one, and here the strength of linkage is more than this one. It means that if the distance of the genes is inversely proportional to the strength of the linkage, if the distance in between genes is more, then the strength of linkage becomes less. If the distance is very less, then the strength of linkage is very more. Linkage, this is a deviation of Mendelian law of independent assortment. This is a deviation of second law of Mendel. Second law of Mendel, this is the law of independent assortment. Mendel's formulate the second law, the law of independent assortment. In case of Mendel, if the genes capital A and the small b present in different chromosome, then the law of independent assortment is applicable. At that time, the capital A gene and the small b gene independent to each other. They assort independently. But in this case, the same if the different genes present on the same chromosome at that time, the capital A and small b, they are not independent to each other. They are linked to each other. They depend to each other. That's why the law of independent assortment is not applicable here. So at the time of inheritance, at the time of transmission, both the genes transmit along with the movement of chromosome. That's why it is known as the deviation of second law of Mendel, that is the law of independent assortment. Now, here we are trying to understand the meaning of linkage with the experiment of Batson and Punnett. Batson and Punnett conducted an experiment on sweet pea plant in the year 1906. Sweet pea plant. They considered two characters. One is the color of flower, and another is the shape of pollen. He crossed two varieties of plant having blue flower, which is dominant to red flower, and another character which is the long pollen, which is dominant to round pollen. So in case of blue flower, I representing capital BB and long pollen capital LL. The red flower, small bb recessive and small ll round pollen. This is the parent generation, and in case of F1 generation, they got capital B small b, capital L small l. So they got all offspring were blue and long pollen, having blue and long pollen. They did the test cross. Test cross means crossed in between F1 offspring with the recessive parents. So this is the double recessive parent, which is capital BB small ll. So the gamete will be small b small l, and here the gamete will be capital B capital L, capital B small b capital L, and the small b small l. So these are the four types of gametes. So these are the four types of gamete from F1 offspring. So these are the four gametes. These are the four gametes of the F1 offspring, and this is the gamete of the double recessive parent. This is the double recessive parent, and this is the gamete of the double recessive parent. What will be the F2 generation? Capital B small b capital L small l, and it will be capital B small ll, it will be small bb capital L small l, and it will be small bb small ll. They astonished with the result of the F2 generation. The F2 generation here, they got blue and long pollen, they got red flower and round pollen, and to another result, these results known as the non-parental result, because blue long, the parent is the blue and long, and another parent having red and round. But here two other characters are developed. One is the blue and round, blue and round. Another is the red and long. So these two are known as the non-parental character. So having one blue here and another round here, this is known as the non-parental. Here one is the red here and another is the long here. So this is known as the non-parental character. And the ratio was 7:1:1:7. Batson and Punnett, they got the ratio 7:1:1:7. This is the phenotypic ratio. So here the frequency of parental character, which is 7 times more than the non-parental character. And Batson and Punnett argued that the parental character, which is 7 times more and the frequency is 7 times more, it means that the character blue and long, the character which comes from the single parent, enter into the single gamete. Red and round, which comes from the one parent and enter into the same gamete. That's why there is the production of more frequency of the parental character than the non-parental character. And they argued that this is known as the coupling theory. And this is known as the coupling theory, which is known as coupling theory. And the non-parental character, one is the, here is the blue and here is the round, blue and round. This is the one parent having the blue and another is the round. The blue and round, these are the non-parental character. The character which comes from the different parent, one is the blue, another is the round, comes from different parent and they enter into different gametes. That's why there is the low frequency of the non-parental character. This is the argument of Batson and Punnett. That's why they argued that this is the repulsive theory or the repulsion theory. This is the repulsion theory. This is the repulsion theory. So according to Batson and Punnett, after the experiment, they argued that there is the two theory. One is the coupling theory and another is the repulsion theory. Coupling theory means if the characters comes from the single parent, they enter into the same gamete. That's why there is the high production, high frequency, high number of parental character will develop in F2 generation. And in F2 generation, very less number of non-parental characters developed because of the repulsion theory. The character comes from different parent, enter into different gametes. That's why there is the low frequency of the non-parental character. This is the argument of Batson and the Punnett. So what is the view, the Morgan's view on linkage? So now come to the Morgan views on linkage. Morgan's view on linkage. Morgan proposed his views on linkage in the year 1910. So 1910. And Morgan proposed his views or developed his views on linkage on the basis of the experiment on Drosophila. So the Morgan did his experiment on Drosophila. This is the fruit fly. Drosophila, and he proposed that the eye color of Drosophila and the shape of wing of Drosophila, these are linked to each other. So both the genes, the eye color gene and the shape of wing, the gene, these are the linked to each other. So according to Morgan views, the coupling theory and the repulsion theory, which was proposed by Batson and Punnett in the year 1906, Morgan in the year 1910 proposed that the coupling theory and the repulsion theory, these are the two aspects of one event which is known as linkage. According to Morgan, the same chromosome or the single chromosome having different genes, suppose that A, B, C, D, the same chromosome, on the genes present in the same chromosome, these are linked to each other, and these genes are known as linked gene. These linked genes, at the time of inheritance, at the time of movement of chromosome, all the genes moved along with the movement of chromosome. So they moved togetherly or inherent togetherly. And here the character which is controlled by the linked gene, these are known as the linked character. For example, in case of Drosophila, the eye color and the shape of wing, these are the linked character. Those genes present on the single chromosome, they form a single group. Suppose that this chromosome which contains A, B, C, D, and these genes which form a single group. So chromosome present in a pair, in a homologous pair, so single pair. So this pair of chromosome which form a single group. So in case of human, we have 23 pair of chromosomes. So we have 23 linkage group. In case of Drosophila, there is the 4 pairs of chromosome. That's why linkage group is here 4 linkage group. In case of pea plant, there is the 7 pairs of chromosome. That's why here the linkage group is 7 in number. So those genes present on the same chromosome and they form a single group, and the chromosome present obviously in a pair in case of diploid organism. So one pair of chromosome means one group of linked gene, one linkage group. In case of human, that is the 23 pairs of chromosome, it means that 23 linkage group we have. In case of Drosophila, that is the 4 linkage group. In case of pea plant, that is the 7 number of linkage group. Now come to the chromosome theory of linkage. What is the meaning of chromosome theory of linkage? The chromosome theory of linkage, which was proposed by Morgan and Castell. Morgan and Castell proposed the chromosome theory of linkage. And what are the different postulates of chromosome theory of linkage? So according to this theory, all the linked genes, they are present on the same chromosome. So all the genes, those shows the linkage behavior, they are present on the same chromosome. present on the same chromosome. Suppose that here A gene and B gene, suppose that this is the color of flower A and this is the height of plant, these are linked to each other, both are present in the same chromosome and they shows the linked behavior. And according to this theory, these linked genes, they are present in a linear fashion on the chromosome. So genes are present in a linear fashion on the chromosome. Another postulate is the distance between two genes, which is inversely proportional to the linkage, the strength of linkage. So the distance between two genes, which is inversely proportional with the strength of linkage. If the distance is more, then the strength of linkage is less. If the distance is very less, then the strength of the linkage will be more. And the linked genes, they tend to remain in original position. Original position. So the linked gene always tend to remain in original position at the time of inheritance. And the linked genes, they shows the two type of arrangement. They shows the cis or trans arrangement. Cis or trans arrangement. These are the different postulates of the chromosome theory of linkage, which was proposed by Morgan and the Castell. Those genes shows the linkage behavior, they should present on the same chromosome, and these genes present on the chromosome in a linear fashion, and the strength of linkage, which is linked to the distance between the gene. The distance between the gene, which is inversely proportional to the strength of the linkage. If the distance is more, then the strength is very less. If the distance is very less, then the strength of the linkage will be more. And here, the linked gene, they tend to remain in original position at the time of inheritance. And these linked genes, they arranged in two fashion. Means there is the cis or the trans arrangement of linked gene. So linked gene, they are arranged in a two fashion. It may arrange in two fashion. One is the cis and the trans arrangement. So I have the separate video regarding the cis and the trans arrangement. Now come to the types of linkage. So types of linkage. The linkage, it may be two types. One is complete, another may be the incomplete. Incomplete. If the linked genes, suppose that in case of Drosophila, the red eye color and the vestigial wing, which transmit from parental generation to F1 generation, and from F1 to F2 generation, and from F2 to F3 generation. Suppose that these are the two linked characters. One is the red eye color, and the vestigial wing. These are the two linked character. If these two linked character, which transmit 2 to 3 generation successfully, then which is known as the complete linkage. So the red eye color and the vestigial wing, suppose that it transmit from parent to F1 generation, and from F1 to F2 generation, and again from F2 to F3 generation. So successfully it transmit from 2 to 3 generation, which is known as the complete linkage. In case of incomplete linkage, there is no movement of the linked gene one generation to another generation, because of the crossing over there is the production of the non-parental character, and which is known as the incomplete linkage, the linked gene not inherent, not transmit from one generation to another generation togetherly. The linkage process which inhibit the recombination in a natural process. This is the significance of linkage. Thank you for watching.